Are gram-positive and gram-negative differentiated based on their impact on us? Well, no. They're actually differentiated by differences in their cell walls. Bacterial cells have an extra layer surrounding their plasma membrane. This helps bacterial cells with osmotic stress, preventing them from shriveling too much or exploding. The cell wall also helps in pathogenicity and is a target for many antibiotics, as human cells do not have a cell wall. The cell wall is made from polymer strands of sugar and amino acids called the peptidoglycan. The strands are linked to each other by peptide links, and these differ between gram-negative and gram-positive bacteria. We differentiate between gram-positive and gram-negative cells based on differences in their cell walls. Gram-positive cells have a thick peptidoglycan layer and teichoic acids in the peptidoglycan layer. On the contrary, gram-negatives have a thin peptidoglycan layer and a second membrane surrounding the cell. At the surface of the second membrane, there are lipopolysaccharides. The gram stain is the technique used to differentiate between gram-positive and gram-negative cells. You start off by putting your bacterial cells on a microscope slide. You then apply your primary stain. You then add a mordant, which increases interactions between the dye and the cells. The decolorizing step is used to remove the crystal violet dye from the cells, and this is the most critical step in all of the gram stain. A secondary stain is then used. When looking under the microscope, gram-positive will appear purple and gram-negative will appear pink. The reason for this is that the crystal violet dye is trapped in the thick peptidoglycan of gram-positive bacteria, while the crystal violet dye is washed off from a thin peptidoglycan of gram-negative bacteria, so when the secondary stain is applied, only gram-negatives get dyed. However, it should be noted that some bacteria do not stain according to their cell wall composition. An example of this are acid-fast cells, which stain purple but are actually gram-negative. The gram stain can be useful in the medical field as it can allow for identification of dangerous pathogenic bacteria, along with other tests, to help diagnose patients as quickly as possible and give them the right treatment. Thank you for watching, and I'll see you in the next video. Don't forget to check out these other videos if you enjoyed this one.